Hello everyone, my name is David Robitaille from the Chemical Engineering program at the University of Connecticut. Today we're going to be talking about the effectiveness of polyamidoamine or PAMAM Dendrimers in nanoscale therapeutics for glioma tumors. Today's talk is based on the AICHU paper listed at the bottom of the screen. Just to lay the framework for today's talk, I'm going to first talk about nanoscale therapeutics. I'll tell you what they are and give you some examples of them. Then I'm going to talk about glioma tumors and tell you about some of the challenges that they pose to traditional drug delivery. Then I'm really going to focus in on PAMAM Dendrimers. I'll tell you about some of the characteristics that make them especially suited to be drug delivery vectors for glioma tumors. Finally, I'll finish off by talking about some of the challenges that chemical engineers still face as they try to develop these dendrimers into effective drug delivery vectors. So what exactly are nanoscale therapeutics? Well, based on my research, the definition I came up with is using particles, also called drug delivery vectors, roughly 1 to 200 nanometers in size to transport therapeutic substances like chemotherapy or gene therapy to a targeted area of the body. Now as you look on the table off on the right, you can see that in the first row, Doxil, or in the third row, Oncospar and Pegasus, these nanoscale therapeutics are actually already FDA approved. However, as you look towards the bottom of the table, you can see that PAMAM dendrimers are still preclinical in development, which means that chemical engineers still have a lot of work to do on these dendrimers before they can be released as safe and effective drug delivery vectors. So now, what are glioma tumors? Well, first of all, gliomas are primary brain tumors, which means that the tumor originates in the brain. Second, gliomas are very lethal tumors. People diagnosed with gliomas are often given under 5 years to live, sometimes even under 1, so they are very lethal tumors. Now, what are some of the challenges that gliomas pose to drug delivery? Well, first of all, there's the blood-brain barrier, a physical barrier in the brain that prevents large drug molecules from entering into the brain tissue. Second, there's poor vascularization of the tumor site. Because there aren't very many healthy vessels that go to the tumor region, it's hard for doctors to send drugs to that region. Third, there's mutations to normal cell functions. This prevents scientists from being able to tell exactly how the drugs will interact with the cell. And fourth, there's close proximity of the tumor to crucial brain tissue, which means that doctors have to make sure that the drug only hurts the cancer cells and not the healthy brain tissue surrounding it. So now that we've laid the framework for our talk, let's talk about PAMAM dendrimers and their characteristics. First of all, you can see here from this picture that PAMAM dendrimers are highly branched molecules, which makes it easy to bind a lot of different chemical groups to the outside of the molecule. Second, each generation doubles the number of binding sites. As you can see here from this animation, each time you add a generation or a fork in the branch to the molecule, it doubles the number of available binding sites. Third, PAMAM dendrimers are typically small molecules, about 1 to 14 nanometers in size based on which generation they are. Fourth, due to their chemistry, these PAMAM dendrimers have a hydrophilic exterior with a hydrophobic interior. I'll explain the significance of that in a moment. And fifth, due to all the amine groups on the outside of this polyamidoamine dendrimer, when it's dissolved in water, these dendrimers have a slightly cationic exterior, and because of that positive charge, they're able to bind to negatively charged cell membranes and enter cells via endocytosis. So now let's take a look at why the PAMAM dendrimer is particularly suited to be a drug delivery vector for glioma tumors. First of all, the hydrophilic exterior of the PAMAM dendrimer makes it soluble in blood. Now, because a lot of chemotherapy treatments are hydrophobic, it's hard for doctors to send those drugs through the bloodstream to the tumor site. But because this PAMAM dendrimer has a hydrophobic interior, scientists can now bind the drug inside of the dendrimer and then send the whole dendrimer drug complex through the bloodstream to the tumor site. The second feature of the PAMAM dendrimers is the fact that they have a lot of binding sites around the outside of the molecule. This allows chemical engineers to attach a lot of different molecules, like these targeting ligands, to the dendrimer. Now, as you can see on the right, the dendrimer and the other biomolecule with a targeting ligand have about a 50-50 chance of attaching to this normal cell. However, on the left, where cancer cells overexpress those ligand receptors, the dendrimers have a greater chance of attaching to the cancer cell. This accomplishes one of the greatest goals in drug delivery research, that of targeted drug delivery. Some other notable features of the PAMAM dendrimer are its small size and cationic charge. Now, as you can see here from this picture, a lot of the blood vessels surrounding the cancer cells are actually leaky or porous. So because of the dendrimer's small size, it's able to escape those porous blood vessels and then because of its positive cationic charge, it's able to attach to the negatively charged cell membrane and then enter the cell by endocytosis. This ensures that the drugs get into the cell instead of, as you can see on the upper right of the picture, where free drugs are pumped into the cell and then pumped right back out by the efflux pump effect. A final feature of the PAMAM dendrimer is its generational effects. Because it's relatively easy and predictable to add generations to the dendrimers, the researchers from the study that these pictures came from were able to determine the optimum size and blood plasma half-life times of the dendrimer. You can see here from these images that they found that the 5th generation accumulated the best within the glioma tumors. And that was all due to the fact that dendrimers grow predictably in generations. Chemical engineers, however, still do face some challenges with the PAMAM dendrimers. First of all, the cationic nature of the dendrimer is actually toxic to some cells, so the chemical engineers still have to figure out a way to reduce this cytotoxicity. Second, we have to figure out a way to enhance the targeted release of these dendrimers so that they are only releasing their drugs to the unhealthy cancerous cells. Third, we still have to figure out a way to streamline the cost and method of production of these dendrimers, since they are still rather expensive and difficult to produce in sizable quantities. So in review, we first defined nanotherapeutics. Then we talked about the challenges of glioma tumors. Third, we focused in on the characteristics of PAMAM dendrimers that enables them to be good drug delivery vectors for glioma tumors. And we finished off with the challenges that chemical engineers still have with PAMAM dendrimers. I hope you enjoyed today's talk, and thanks for watching.